Again, I am Art Edison. I'm the director of our CCRC NMR facility and the local PI at UGA for the Network for Advanced NMR, which is funded, you're going to hear several times during this workshop, by the National Science Foundation. So none of this would be possible without the funding from the NSF, and we're always grateful for that. So first, a little bit of logistics. Some of this you already know because you're here. So all times, so you've seen, I think that all of you have seen schedules, or you will not have been here either. But all of the times posted in the schedules are Eastern US time zone. So we're every day from now until Friday, we're going to be starting at noon, and we're roughly going to go until about 4 in the afternoon with some breaks in between. And the whole thing is structured in a consistent format, so that we're going to have from 12 noon to 2 p.m. there's going to be content delivered to you. So we're going to have lecture and some demo time, and all of this is going to be captured by the YouTube live stream that's going right now, and that you all should have links to. Then after that, so you'll be able to go back to that live stream. It's a private, it's an unlisted live stream, so it's not out there and searchable, but anybody who has a link will be able to get on to it and access this material anytime. Eventually, we're going to be editing these things and making them publicly available when we cut them down to size. And then the second 2 hours of each day are meant to be hands-on, and we're just for logistics, and also so that you can feel comfortable asking any kind of question. We're not going to be recording the second set of the time. So from 2 to 4, that'll be just through Zoom. And you'll have to have your NMR box account set up by that time, or you can still watch live. You can still watch what's going on in the hands-on, but you won't be able to do hands-on. And so we'll talk about the NMR box in a minute. So all of the sessions are going to be through this same Zoom link that you just got on right now, so every single day you go to the same Zoom link. And then as I think that a lot of you are on Slack, so we have a few different options for questions and feedback. You can share chat questions along the way. If given time at the end of talks, we're going to open it up for a few verbal questions. But you can also put questions and answers and have a general discussion on the Slack channel that we enrolled all of you in. So that's really the most enduring and probably the easiest, because we can do that over time after the workshop is ended. And then the live stream is there, and you can post comments and questions there, but they're not going to be dealt with on an immediate basis. So the link each day is going to be a separate YouTube link, a live stream link. We can't have the same link the whole time. So every day we will communicate with you where to find that YouTube live stream, and then so you can always capture the content. And we're going to talk a lot more today about the NMR box account. I know that a lot of you have had some difficulty getting your NMR box account set up, but I think many of you have been successful now. And Mark Macheski, who is really the architect of NMR Box, is here to tell you not only how it works, but to help you in the hands-on session for troubleshooting. And then select participants, we had to be pretty selective about this, are going to be able to do hands-on NMR data collection. When I say hands-on, we're setting up virtual access to our spectrometers. But everybody is welcome to just watch the sessions. You'll still learn something, even if you're not pushing the buttons. You'll still learn about operating the spectrometer. So you're always going to be welcome to join those. Okay, those are logistics. This is the team of people that are really instrumental in this particular workshop. Everybody now knows that you wouldn't be here without Karen Howard. She's on this Zoom right now. Frank Delaglio at the National Institutes of Standards and Technology will be our speaker on Wednesday, talking about advanced NMR data processing. He's the author of the very famous and very well used processing program called NMR pipe. He's going to be showing you how to do NMR pipe processing, talking about processing NMR data in general, and then introducing something that we've published with Frank called SAND. John Glushka is the person who is really going to be leading the access to the NMR spectrometers, and so especially the 23 participants who have been picked to have some NMR time after this. They're going to be working with John today from starting about 2 o'clock to get on. Laura is our technical lead. She's going to be helping with all of the problems that come up. Mario Uchimiya is the metabolomics specialist in NAN who is really developing all of the materials that in the KB, and he's going to take the lead on showing you some work on C13 towards the end on the last day of the session. I already mentioned Mark Macheski at UConn. He's not only part of NAN, but also NMR box, and he will be helping us understand what's possible in NAN. Conrad Epps is in my lab, and he is working with fraction libraries, and he's going to be one of the people who will be helping in the Thursday session when we get to that point in hands-on. Chi Cheng at UConn is the whole project manager of this Operation NAN, and without her, none of it would be happening. John Grimes is another person at UGA who is involved with NMR data collection. He's our facility manager. He and John Glushka will be helping you learn how to do NMR. Chris Asselman is another student in my lab who's going to be demonstrating fraction library work. Okay, so those are the main people on this. There are a couple of others. In fact, I remembered I didn't put Zarif Hussein, who will also be joining the fraction library help. So this is the schedules. I should also say, I'm going to show you where to find these slides at the end of this presentation. So you don't, so these slides are all available if you get your NMR box account, and I'll show you where. So we're here right now, the intro to NAN. Mark is going to introduce some of the features in NAN that will integrate what we're collecting on the NMR data or NMR spectrometers and integrate that with NMR box. And then he'll be giving both slides and also a demo of capabilities. There will be an opportunity for question and answer. We'll take a short break. And then this afternoon, we will have two breakout rooms. Mark is going to be in one, and you can ask him questions about NMR box related issues, or he can even help you get signed up if you're not in there yet. And then John Glushka will be in another one, and he's going to be helping the 23 people who are going to get NMR time signed up so they can run the spectrometers. Tomorrow, we're going to start same time, 12 noon. I'm going to give an introduction to metabolomics as a field in general. These are very broad, short introductions, and then I'll just briefly introduce NMR spectroscopy. John, at that point, will give a demo of NMR to all of you and just show some, how, collecting data on our instruments. There will be question and answer break, and then we'll have a hands-on session. This is particularly useful. This is required if you're going to be if you've been given NMR time to try out at the end, to go on and get some experience with this, but it'll be really useful for anybody who hasn't had NMR experience up to now. We'll open up a couple of our spectrometers, and both John Glushka and John Grimes will be running these things with you. On the third day, Frank Delaglio is going to give the lecture and then introduce this thing called SAND, which is a time domain NMR analysis that really, in our opinion, really simplifies what we can do in metabolomics. So we think it's a great resource that people with doing metabolomics by NMR should know about. And then we're going to get a demo of SAND on NMR box that are going to be facilitated by Leandro, Chris, and Frank. And I think I also forgot to put Leandro on the intro, so another student is going to help with a lot of this. And then we'll have question answer break, and then during the session from 2 to 4. Then we're going to have 3 different breakout rooms, and Frank is going to go from breakout room to breakout room and just see how things are going. But all of you will have a chance to use NMR box to apply SAND processing to your own data or to data that we have on hand for you to try out. Day 4 is fraction libraries, where we're going to show you a concept that's really not new. It's a pretty old concept, but we're going to talk about why we think that fraction libraries are very useful for metabolomics. We'll get that's a good opportunity to bring automation into it. So John Grimes is going to be giving a demo of using Icon NMR and Brooker to collect a series of samples automatically. Chris in my lab will be demonstrating batch processing and some analysis of fraction libraries. And then the afternoon breakout session, it'll either be going to NMR box to analyze data or to the Brooker to work on collecting data that afternoon. And then the last day is C13 NMR metabolomics. That is another thing that is a real favorite of ours. I'll be talking about why we're doing that. John Glushka will demonstrate some collecting of the data, and then Mario is going to show you some of the tools and resources that we've been developing in our lab to analyze C13 data. And the afternoon we'll have those 2 breakout sessions as well. For data collection and analysis. And so I hope that tomorrow you're going to understand why we're putting these general topics together. So we have fraction libraries, we have SAND analysis, and we have C13 NMR. And I'm going to make it more clear tomorrow why these all make sense in this context to go together. But essentially, it's to pull out more efficiently information from crowded, overlapped NMR spectra. So just to introduce NAN in general, the Network for Advanced NMR was started by these 4 PIs. Jeff Hoke at the University of Connecticut is the lead PI. UConn is the lead institution for this, and they form the hub of a network right now of just these 3 institutions, but we hope to add more. I'm in charge of the group at University of Georgia, and then Chad Reenstra and Katie Hensler-Wildman are up at the University of Wisconsin in Madison. So we all came together to form this virtual network and to try to make it so that our overarching theme is democratizing NMR. To make it easier to do NMR for people who are not experts. And this whole thing is funded by the National Science Foundation through a large major research infrastructure grant that I'll tell you a little bit more about in a minute. So what is NAN? It's a bunch of different things, and I'm going to try to give you a sense about the way we think about it. It's a place for biomedical researchers to go, or it could be for material science too, if you connect with the Wisconsin group, to learn about NMR and how it might be useful or might not be useful for your project. It's a place for NMR experts to share best practices, to try to share ideas that they have and protocols to perform different types of NMR experiments. It's a place where you can go, we hope, to find resources, both instruments, what might be out there and available for research that you're doing, and expertise that you would need to carry out a particular product. I guess the overriding principle is that we don't all need to have the highest field NMR in our own lab all the time to be able to use these kind of resources. We take particular pride in what we've been managing to do recently in data. And so the data management is a big part of NAN. Mark is going to tell you a lot about it. So we're basically going to be archiving and storing and making available to the public both raw and processed NMR data. It's going to be linked with sample information and how the data were actually acquired and processed. And then a network of the different capabilities of state-of-the-art NMR spectrometers that have a bunch of different capabilities that are available through NAN. So the collective is much greater than any lab can have on their own. And so if you were a novice in NMR, which I think many of you probably are, these are the kind of scenarios that we envision somebody coming up to us or approaching getting into NAN. The idea is that, so if somebody says, I heard somewhere or read in a paper that NMR might be useful to solve a problem that I'm working on, but I'm not sure where to even start. Then we hope that you can find NAN and sort of dig in and find some resources that could give you some clues, and contact us too. Is NMR the best method to answer my question? Or if I need access to NMR equipment and I need to get access to NMR equipment because we don't have that at our home institution, and I also need help acquiring and processing the data. That's really a key constituent that we're trying to help. I read a paper and I now think that I need to do an NMR experiment, but I have no idea how to do it. So I think that captures a lot of people who don't know it, but they know that maybe this is useful. So we're trying to make that easier. If you are an NMR expert, we still want you to be participating in NAN. For example, if somebody is studying a new system, whatever it is, proteins or metabolites or materials, what pulse sequence is best for that? Either a new biological sample system or perhaps a different field strength. What pulse sequence is going to work best? How can I collect my data? I need a specialized instrument that I don't have right now that's not available locally. How can I get access to that? I'm looking to connect with collaborators who have biological expertise, and maybe together we can write grants and get our projects funded. So those are the kind of people that we're really targeting for a lot of this. So I'm going to just give you a little bit of a bigger overview of what it's all about in terms of this network. So again, it's funded by the NSF, a Mid-Scale Research Infrastructure 2 grant that we got about 3 years ago now. The hub is UConn, and you're going to hear from Mark about NMR Box and the resources at UConn to collect the data and organize the data. And then our colleagues in Wisconsin have a large NMR facility in Madison called NMR FAM. It's actually where I got my PhD, so that's a nice thing for me to be working with them again. But they have really great instrumentation. We both have big facilities with a lot of existing instrumentation. And then Wisconsin installed a 1.1 gigahertz spectrometer that's been working for about 6 months or so now. It's actually over here in this corner of where my laser pointer is. So that is up and running and collecting really useful data on solid state NMR. This is our facility at the CCRC, the Complex Carbohydrate Research Center at the University of Georgia. And we have just gotten our 1.1 instrument, and it's at liquid nitrogen temperatures now. So we've got about a month or so before it's fully energized to a spectrometer. That's actually the reason why we're doing this virtual workshop, because it was just too chaotic to try to figure out how to coordinate with the installation. So again, we want the founding principle is really democratize NMR, make it easier for people to use. We want to try to combine state-of-the-art data tools and secure and simple management tools on NMR Box. We have these two different 1.1 gigahertz magnets that are available to the world. These are not just for our use at Wisconsin and Georgia. So these are for the user community at large. We want it to be easy for expert labs, real NMR labs, to just consider this an extension of your lab. If you need to use one of a field strength or a probe combination that we have that you don't have available, you can send a sample down and we'll give you access to the time. users will send samples. They can send samples to the facility rather than travel, but you're welcome to come on site if you're able to get here and want to. NAN staff can help with experiments, and you can do remote access. We want to make it easier to find things. So we talk about a reservation system. Not all this is built yet. We're still working on some of these final things, but the term that we like is Airbnb. So not Airbnb, but if you're going to go to a different city and you know that you need to be near a certain area, you can search and try to find what's the best place for me to live. So this is what we're trying. We're trying to make it easy for you to find the right NMR resource. NMR data from all of our spectrometers right now, it's up and running just fine. It all gets transferred automatically to NAN and NAN resource connector and eventually to NMR box, and then people can access their data for analysis on NMR box. We're establishing, in fact, you're part of this now through this Slack invite that you all have, and is to get a NAN community network. This is one of the mechanisms that we're going to be using for that. So it's a way to link experts with users with interesting things, question answer. We can make announcements about functionality, et cetera, announcements for workshops like here. And then I would say almost the core component of NAN is not so much the big spectrometers, although those are really exciting and important. It's really that we've been developing knowledge bases for the whole time that we've been funded. And we at UGA we have two knowledge bases, one in metabolomics and one in structural biology, solution structural biology. These are so we're developing tools to be able to, protocols on how to make samples, data on NAN to show you what the data should look like, processing protocols for NMR pipe, whole sequences on our spectrometer to make it simple for you to know how to run and collect the data. At Wisconsin, they have two KBs called one on biosolids, so solid state NMR used for structural biology and also material science. So very complementary they span a lot of NSF's interest in terms of funding. We have annual hands-on workshops. This one this year. We may stick with virtual or we may go back depending on how this works and how all of you like this. But then we have protocol standardization, data analysis, metadata tracking, things like that. Okay. So I've talked about these components. We have Mark is going to be going over the resource discovery, time requests, and access data stewardship kind of tools that we have in here. These are the knowledge bases, but then we also have between all of us, actually is greater now than 19 high field spectrometers. So there's a lot set up right now that is available to users through NAN, including spectrometers at the University of Connecticut. So we already have a network of a lot of different capabilities. And just in the last couple of minutes of my presentation, I want to show, I think that this will start. Yeah, so on the left, this is a video. So this was less than 2 weeks ago, or about 2 weeks-ish or something ago at this point. A little bit more of our magnet, this 1.1 gigahertz magnet being put into our facility. So this is a crane. You can see the scope of this and the people that were up on the top of that building. This is where our NMR facility is kept. And so this is just sort of a dramatic view of the crane taking the hatch off. And then in this middle photo is a nice picture of the crane lowering the magnet into our facility. And then on the next slide, this was actually a live stream that we generated during the installation. This is a twice 2x speed of this thing coming down. It was obviously done very slowly and carefully, but so that magnet, just to put it into context, that's about 8 tons and being lowered very slowly, 8 tons, and also over $10 million worth of a magnet being lowered gently onto our floor. And then this rigging that's over here on the left is where the magnet is being set up right now and being cooled down. So anyway, you can look at this whole video. We have an entire live stream of the whole process if you want to spend 4.5 agonizing hours just watching people go back and forth for this short little moment. Okay, I'll let you go through and watch that coming to completion. This is just an interesting view. This is our facility 5 years ago on July 10th, or 2019. This is what it looked like. So we have a 900 at one end of the hall. We have an 800 with a sample changer on the other end. We have several different 600s. At the time, we had this little 300. This was an old 500 that was not very useful. And this was an unshielded 600. And then this is what it was just a few days ago. Actually, that this platform is finished beautifully right now. And so the 1.1 is right in the center. It's just been the finished cooling to liquid nitrogen temperatures, and we're going to start cooling it to liquid helium temperatures in a couple of days. So that's what our facility status is. And now, just briefly before I turn it over to Mark, I want to show you this is sort of a high-level overview of the functionality that we have tried to incorporate into the design of NAN. And you're going to hear descriptions of many of these things from Mark at the end of this presentation. So this Air B0 resource discovery, how do you search for equipment and expertise? Where do you go to find protocols? Putting in time requests for the spectrometers. There's a data browser where you can try to find data that are in there that are publicly available. And we want to try to make this into a really useful NMR raw data resource so that things like machine learning can take place more efficiently than we could do in the past. We have sample metadata that can be associated with instrument data. The instrument data is actually harvested from our spectrometers through this thing called an automatic data transport. You'll see an example of that. And actually, John is going to show you some of that tomorrow in a demo. So it's working. It's pulled from our spectrometers. And then getting the processing pipelines to analyze data in NMR box. And then BMRB, BioMagRes database, is a repository that's associated with this whole NMR hub, we call it, that's part NAN and NMR box and BMRB. And we want to try to make it easy for people to deposit your data and to use the BMRB resources. So in summary, NAN, we think, can really help you with your research. Our goal is to try to enable more people to use NMR in their research. There's a wide variety of NMR spectrometers that are available. You can come here or you can use remotely. Data infrastructure is really one of the highlights of the whole thing. It makes it easier to manage your data. And we want to build up even more the network of experts and resources for you to solve problems. And so we've reached a point where this data infrastructure is built out enough that we will be able to start adding to this network. We hope that this network grows. We don't want it to be an exclusive club. We think that the bigger it gets, the more useful it will be. And we also want people outside of our institutions to really start utilizing this resource.